I'm sure that we would all agree that maybe this is not a smart idea just to hit somebody in this context. So we can think about it. Our parents would also agree with us and teach us that that's not the right way to behave. But I think we also have collected some data during these years that suggests that there is another piece of information that could play a role in these big puzzles of decision making, which might come indeed from the fact that when we observe somebody is in pain or in distress or in an emotional state, and we can, even if we just imagine that person being in that situation, we activate brain regions that are normally active when we experience that feeling, that state too, right? So this, as I said, I don't want to already, this is a defensive beginning of the talk, which is probably not good, but does not explain probably the whole story. As I said, it's just one of the piece of information that our decision-making system might use in order to actually decide not to hurt the other person. So as Christian anticipated yesterday a little bit, there is a lot of work we've been running inspired by the work on monkey and mirror neurons, in which we indeed identify these brain regions that are activated in both the first-hand experience of an action, for instance, and when you observe somebody else performing an action. So we would look for area activated while you perform an action and areas that are activated when you see somebody else performing an action, and then we would simply look for the overlap. Boxes activated in both cases. And then because these areas fall in the mirror regions, and where we have neuronal recording showing that the same cells is activated in both cases, we have this idea that we perceive the actions of others because we basically bring them in our own reality. We use the machinery that actually makes us act. We embody the other person. And as Andy also anticipated a little bit, we do have a similar mechanism in the case of touch. When we are touched, we activate some of the sensory regions. When we observe somebody else being touched, we again activate the same regions in a nice overlap. And now we know, thanks to his work, that this is true also very early on. It's not just an adult result. And similarly, for the case of emotions, we have regions like the anterior insula that is activated both when you experience an emotion, in this case was disgust, and when you observe somebody else being in disgust. But this is true not just when you see stimuli. For instance, also in the case of actions, it's not just a visual input that is the only one tapping into, for instance, our premotor cortices or the insula. The sound of an action is also a modality that would work and would generate the same overlap. So it's not specific to vision, the way in which the signal comes in. And if you read a text like this, when you turn around to look who is leaning on your shoulder, you are peering into the unsightly face of a homeless guy. The guy leans forward and discharges the complete content of his inflamed stomach on you. You're covered with decaying vomit and so far and so forth. It can get only worse. And again, just by reading the text, you would have an overlap in the anterior insula. And the anterior insula will not only show an overlap for disgust, will also show an overlap in the case of happy or more positive stimuli. It would show an activity and an overlap for pain and sadness. So here, there could be that if we zoom in at the level of cellular level, we would find neurons that are more specific for each of these emotions. And in a way, the work that Chris showed yesterday in the animal does suggest that. When he found these neurons, for instance, that are mirror for pain and the ones that are mirror for fear, but they were all colocalized in the ACC, but then the specialization happens within that region. It could happen here, but we don't know. So the work that he ran, or we are running together with him, was done on the anterior cingulate cortex. This is because that area is much more preserved between species than the anterior insula is. And so what the anterior insula does, it's still a question mark, and it's very challenging to me, and I hope one day to be able to tap into that region. But so far, the only thing that we probably convincingly showed with the animal work is that the reactivation of our own emotions or sensations or actions influence the way we feel. So the rat was when the moment in which we block this, the anterior cingulate, the rat would stop freezing. So the motion was not contagious, but the motion of the other as well as it was before. But does it mean that it was also perceiving what was going on in the other rat? And this is a bit more difficult to test in the rodent, but we can try to test it at least in humans. So the approach that we've been implementing is obviously, as you can anticipate, is by using TMS, right? I guess everyone knows what TMS is, no? Yeah? Okay. With TMS, it's basically with a magnetic field, that is the transcranial magnetic field. So you have a tool that produces a magnetic field. These magnetic fields, as a consequence, will produce a current, and this current will interfere with the activity of the neurons that are below that surface. So the idea is that you can figure out whether the activity of a certain region is or not causally involved in a certain behavior. So as if you're an alien and comes to Earth, it's a very simple concept, and you go to the highway, every time you see the light of a car, the car brakes. So if you don't know that it's the person inside the car that makes the car stopping, you might assume that the car stops because the light was on. But then you have a very easy way to test it. You can just break the light and see whether the car still stops or not. And the idea of using TMS is the same. TMS can interfere with the activity of the neurons just below. And then if you observe a change in behavior, then you can conclude that the activity that you perturb was involved in that behavior. But before going that way, we also need to go away from group analysis. Because, okay, if we find an overlap between experience and observation at the group level, what does it mean for each individual? If you want to assume or say that that activity helps us to perceive somebody else, then that has to be true within each of us. So what we've done at first was to rerun our analysis at the level of single subjects. So this is the activity that is activated both by the experience, observation, of action. And then you can simply threshold this activity and attribute a value of 1 when it's significant and a value of 0 when it's not. And then you can sum up all these maps, and what you see here is a consistency map. So the colors this time indicate how much a particular voxel or how many individuals have this overlap in that particular voxel. Okay? So here we have that the majority of our participants had activation in these voxels here in the somatosensory cortex. I mean, every participant had voxels in the circuits, but then the somatosensory was the area that resulted more consistent through our individuals. And that was a bit surprising because this is an action study, and at that point we were mainly focused on the premotor cortex. And so we're like, okay, why the somatosensory is there? What could be the function that it has? And let's see. Ruth, can you pass me that glass? Okay, is it empty or full? Empty. You can deduce how heavy this object is by simply watching me lifting it, right? And so what I want to say is that an action is much more rich than simply containing movement. You know, what it conveys is also the many more properties, how the surface is, depending by how I grasp it, and how heavy the object is. So maybe, given that when we perform actions, our motor and somatosensory system is so well connected, and we need both in order to really precisely act in the environment, we thought, oh, that's why the somatosensory is there. It is also helping us to add all these attribution of the action. And here what I'm simply showing is that most of these areas were indeed, some of these areas were considered the homologue or where the mirror neurons were found, so like the ventropremotor and the IPL. But then MRI gives also the opportunity to explore other areas. And as I said, we had a couple of areas between the somatosensory cortex and the cerebellum that were not expected at first, but it makes very much sense. So my main question, because at one point I really got a bit bored of just seeing blobs. I really wanted to understand whether this blob had any meaning to our everyday life, so to our perception, to our behavior. And as I mentioned before, we use TMS to investigate this question. And the somatosensory cortex is very superficial on the head, so that makes this tool very useful because the TMS cannot really go, cannot precisely stimulate regions that are deep into the brain without also stimulating all the areas above. And, but the somatosensory, being superficial, is a perfect target for that. So if we think that the somatosensory is helping us in perceiving for the weight of the glass, right? If I now go and perturb this region while you're asked to indeed estimate the weight of these objects or to tell me which of these two objects was the heaviest one, then if it really plays a role, I would expect a reduction in the behavior, like in the task performance. And this is indeed what we observed. When we disrupt the activity of this region, participants were worse at telling us which of these two objects were the heaviest one. And then we thought, okay, this is kind of nice. Then we do use the information in the somatosensory to interpret the actions of other people. But is it specific to observing a human action, a biological motion, or the somatosensory is just giving me the weight of everything in the world? So we had another task in which we had balls falling down, and then the participants had to say which of the balls is the heaviest one. And when we do that with TMS on the somatosensory, we did not observe any change in the task performance. And then we went a little bit back because we wanted to make sure that the results were really coming from a stimulation of the somatosensory. And then when we stimulate the posterior parietal behind the somatosensory, no changes in task performance. When we go in front on M1, we also did not observe any change in task performance. So our data at first suggested that the somatosensory between these areas was the only one having an active role. And, but Antonia Hamilton, even before us, they stimulated the premotor cortex. And then again, when you do so, you observe a change in task performance. So now what you see is that if you basically start playing around with regions that belong to the same circuit, then you do find a change in performance. And what about the cerebellum? We saw the cerebellum before as well. Is it also involved because it's part of the circuit? So we had the opportunity to test people that had a spinal cerebellar ataxia, which is a degenerative disease of the Purkinje cells, and it gets worse with time. So when we asked our participants to leave two boxes and tell us which one is the heaviest one, so not to observe, but simply to do it themselves, the patient with the spinal cerebellar ataxia performed worse than the control group. Then when we show them videos of somebody lifting an object, then patients were again worse than the control group, suggesting that a deficit in the cerebellum was also impairing perception of somebody else's action, not just of your own. And the degree of the impairment correlated with how much worse the participants were at the task. So there was a correlation between the severity of the disease and task performance. But then people normally attribute to the cerebellum like the role of analyzing kinematics of movement. That's quite of an accepted statement, I would say. So we decided to, I thought I had it. Let me see. Oh no, because I copied the previous version, sorry. But we had an additional condition in which we covered this arm with a sleeve. And the idea was, okay, if the cerebellum only codes for the kinematic of the movement or gives us the weight of the object through the kinematic, then we should not observe any difference between having an arm covered or not, because the kinematic is the same. But if there is anything additional from the shape of the muscle, because participants were reporting that they were using the shape of the muscle, eye tracking data are also showing that people would look at this point, then if there is something, then we should observe a difference in performance or between the patients and the control that is an interaction with the condition as well, in which then we would expect the shape, a deficit also that is bigger in there. But then what we found is that indeed patients were generally worse than control in all the conditions, but when we do look at the difference between sleeve and not sleeve, which should capture the shape information, so no sleeve minus sleeve, then in that case there was no difference between control and patient. So suggesting that this additional information of shape is not what is computed or so strongly elaborated in the fair value. But then the main message I would like to, that you bring home as well, is that whatever region you stimulate or you perturb that belongs to a circuit causes an impairment in the task, suggesting that what we are perturbing is the entire circuit. So the question is, what are these areas doing? Are they working independently, and then we just perturb each information independently? Or are they integrating the information and working together? The way we tested it was again using TMS. And participants came in, we localized the shared activity, voxels common to action, observation, and execution, and then we did an offline TMS session while they were watching the videos. And here what we did, we took our region of interest, the somatosensory cortex, because that's where we stimulated, and we computed our contrast of interest. So we looked for activity in this region that were responding more to the action observation compared to the control, and more so during the TMS stimulation than the sham. Once you have this value for this region for all your participants, then you can do something like this. If I now take an individual, and if TMS is causing on your a big change on your somatosensory cortex, and I take another voxel that I think is connected functioning to this voxel in the somatosensory, then I should observe a big change in the activity of that voxel too, because they are functionally connected. But then if I take another person, you for instance, for which the TMS didn't alter the activity of the somatosensory so much, then I would expect that if I take any other voxels connected to the somatosensory, I would also observe no change in their activity, because there was no causal link. So you can run this regression whole brain, and basically what you see here in red are all the voxels that change their activity as a consequence of a perturbation of the somatosensory cortex. So what you see, if I now plot the circuits that we mapped on the first day, you see that if I perturb the activity of the somatosensory cortex, this perturbation is sent over to the premotor area and also back to the visual system, suggesting that all these areas are connected to each other functionally. And if you perturb one, you're perturbing really the whole circuit, which challenges also the way we normally interpret the TMS results, where we really conclude, oh yes, the somatosensory, that the causal influence. And yeah, well, yeah, it does have a causal role, but what specifically it does, it's still a question mark. So this was the case for actions and sensation, and for the emotions, as I mentioned, we don't have good tools to go and manipulate the brain activity there, except if we use patients. And we have some results from patient work in the past, where people had lesions in the anterior insula, and what these people showed is not only the fact that they were not able to perceive this gas so well. So if you would offer, so which cake, by the way, would you choose? This one or this? Huh? I guess the chocolate one, right? Yeah, so this patient would choose by chance either of them. So they kind of lose the ability to recognize this gas. They don't feel this gas as we would normally feel. And when you stimulate in these areas, you would also induce this movement of, like you're going to vomit. And these patients also lost the ability to perceive disgust when you show facial expressions of disgust. Again, suggesting that there as well, the insula is involved in the perception of disgust in Alzheimer's. So I hope to have brought you some data suggesting indeed that this activity is involved in perception. And these conclusions was made by the fact that if we perturb the activity over there, we also perturb our perception. And now the next question I would like to go is one step forward in perception. So when we hurt ourselves, if I now kick here, right, this sensation of pain that I perceive is what would motivate me to simply go away, or next time I step to try to stop before I hit it. So it's really like the pain I feel helps me to remove the cause of pain. So if this activity is involved in the perception of pain of others, it could be that this perception also helps us somehow to motivate us not to hurt others, in a similar way we have on our first perception. So the way we decided to investigate is to try to have a more naturalistic task and complex, that's a consequence of the naturalistic task, in the lab. So you would come in and you would meet another person, and then the other person would receive electroshock on the hand, and you would be either with an EEG cap on your head or in the fMRI, depending by the version of the experiment. Then the person that is from which we record the brain activity would have the choice. We would give them like €6, and now the person has the choice to keep all these €6 and then bring them home, or to give up some of these euros in order to reduce the intensity of the next electrical stimulation to the other participant. So this is the other participant, the receiving shock. So you would see her face and her reaction to the shock. And then you have the €6, and now you can think, oh, this is not painful at all, or I want the €6. And then what you would see next is another video that has the same pain intensity as the first one. On the other hand, if you would think, oh, was actually quite painful, poor girl, I'm going to give her €4 and 5, okay. And then the next video that you would see, it will show a stimulation, and the reactions to stimulation that was way lower than the previous one, actually 5 times lower than the other. So it was really a one-to-one relationship between donation and the reduction in intensity. And what we observed, this is what we expected, is that donation and intensity of the video, of the pain shown in the first video, very nicely correlated. So people donated more often, or they donated more money when they perceived more pain. So, as I mentioned you before, I would expect the anterior insula here, or the anterior cingulate cortex, being activated by this stimuli and being part, and I would expect it to correlate with the donation as well. But we don't have a way to test it. So I thought, okay, what can we do about it? Well, what we also know is that when you see the origin of pain, so if you see that I'm hitting myself with a hammer, or you see a cut in myself, I do not only activate the insula and the ACC. I also activate the somatosensory cortex. So I thought, okay, let's give it a try and generate stimuli that explicitly convey pain and the amount of pain by the body part that was affected. And then we do the same task, but with these stimuli and on top of the faces. So what we do here, first of all, we go and take from the G signal the ones that originate from the somatosensory cortex. And then we want to see whether this signal correlates with the intensity of the observed pain. And indeed, this is the case in the somatosensory cortex for both the face and the hand stimuli. So what you see here is that this is a 2 seconds video. So the 0 is the time at which the face starts to show the pain or the hand receives the slap. And here you see the signal that correlates with the intensity, the observed intensity. And you see it for both. This is a bit shorter, but the hand reaction is much faster as well. So it makes quite sense. Then what we did, we used TMS on the somatosensory cortex during the presentation of the first video. And what we observed there is that the relationship I showed you before between donation and the intensity of the pain was basically tilted. Was not as nice as it was before, and people started to donate more randomly than before. But this change in the relationship was only true for the stimuli with the hand. So here the activity was at first correlating with both the face and the hand. But then when we perturb the hand region of the somatosensory cortex, these only affect our perception of the hand stimuli, and we changed the donation only towards this. And we were curious to see whether this change in donation, in the relation with donation, was caused by a change in perception. So when we do stimulate there, is it because we change your perception, or we directly change the way you donate? So we had another task in which we simply asked our participants to rate these movies instead of donate, just tell them how much pain do you see. And in this case, we did TDCS instead of TMS, but the idea is the same. You want to perturb and this region. And when we do this, so you can basically replace the donation with the rating, and you plot it against intensity. Again, when we perturb the somatosensor, we observe a change in this relationship. In this case, it was in the opposite direction, simply because we used an excitatory stimulation instead of an inhibitory. But the change was there. So we kind of concluded that this change, the influence of the somatosensory into the decision-making, was probably mediated by a change in the way we perceive the stimulus. So our donation might also be mediated by the way we perceive the stimulus. So to sum up for this part, so we have the other and the pain of the other, and this taps into the way, the mechanism that make us feel in that way. And I think that one of the reasons why we do this is to allow us to attribute a value to the pain of the other. And why is that important? So in our everyday life, we are mostly, unless we do people watching, which is quite fun once in a while, but in most of our everyday life, we act with others. It's an interactive world, right? And the decision to help or to interact is always a decision that compares like the cost of this helping with the benefits of this helping for myself. But in order to compare costs and benefits, I need to have two entities and two values that are on the same unit. And the pain that I observe in others is yours, it's not mine. How do I know how much pain you have? How can I attribute a value to something that does not happen to me? This is a very difficult computation to do. And if you use the same areas that make you feel in pain to bring the other into your own self, then you have a nice way to compare the value that you attribute to pain to self and the other. And then you can also get the information from other regions that says, well, this is not you, it's the other. And then you can really build up a complex system that helps you to attribute the value and to compare it based on what is at stake at that moment. So the money in case of the selene, or like if your friend stole your partner, how much the value of your friend, or the fact that he stole your partner against his pain, and you can compare it and then take a decision. But it is also true, as Jean pointed out many times, and I totally agree, that we are not always acting prosocially. We are not always nice to each other. Like, what if you're a boxer, for instance? I'm going to go in a more extreme case, even than just annoying each other. Like here, this waiting has a big role, like money play a bigger role than the 20 cents that you would get in my previous task. And the amount of pain that you do to others is way stronger. And many of the boxers would choose for winning the competition, because what's at stake for them is very important. So how is it? Is it that these people don't have this part of the story, so the system is not activated? Or can they simply switch it on and off? Or it's more like a dimmer. We can all regulate this activity based on the context and the situation, and then we have a nice tool that we don't always have to use. So here I'm going to show you quickly a couple of examples that illustrate how many variables can actually modulate the activity in these brain regions. And one of the very first experiments we used to measure perspective taking, how much you're good at putting yourself in the shoes of others. And many of you probably know the interpersonal reactivity scale of Davis, and one of the subscales for measures in this perspective taking with sentences like this, and then you need to give a value from 0 to 4, where 0 basically is when the sentence does not represent you well at all, and 4 when the sentence represents you very well. So when I'm upset at someone, I usually try to put myself in his shoes for a while. Like, who would say 3? Nobody? First time. Normally at least one or two. Who would say like 1? 2? Well, keep it for yourself, okay? And then I will show you why. So here you have a couple of more questions and the same story. And then what we've done here, we mapped the mirror activity for the sound of action and for the group. And then we have the score on this perspective-taking scale of our participants. We took the 6th with the highest score, and we plotted again the activity, and this is what you see. And then we took the 6th with the lowest score, and this is what you see. Okay, this suggests that there could be inter-subject variability in how spontaneously we activate these regions to the observations of others. And another simple case of inter-subject variability also comes from the example of the experiment with the actions again, is that we had the opportunity to scan people that were born without arms. So they were using their foot very properly, very nicely, very skillfully, much more than we would be able to do. They would come and say, hello, I'm Valeria, right? And we wondered at the point, okay, they don't have ever experienced any action with their hand and their arm. How do they perceive the actions of other people when they're made with hands? And here I'm asking you to identify which are these two people. Which are the people that are born without arms, and this is the activity why they watch the hand actions of other people. So, yeah, number 5, exactly. So I've asked this question many times, and normally people suggest number 5 or 7. Huh? 16? Okay. Everywhere. No, yeah, I think that the idea that was suggested is that because they don't have the arm, they probably have a reduced activity in these regions, right? But actually, these two are the guys, so they're perfectly in the middle. They activate everything, and this is when you look at where this happens, it's very much in the hand representation, which is surprising. I mean, they do all the actions I've shown. They do them with the feet, very skillfully. And the feet representation is very measured. And when I plot the feet representation, indeed the feet goes from measure to the hand region. And the mouth representation goes from the mouth to the hand region. But then it's still interesting that all the grasping is still mapped in the center, in the hand representation. Again, suggesting a nice interaction probably between a genetic component and the environment. So other ways to manipulate that have been discussed also yesterday, to manipulate the activity in these regions is just by creating artificial group or to make them play in different occasions. So Tanya Singer did a lot of work on this, and if you, for instance, you make people play with other 2 participants, and so you have 3 females playing, and then 1 is our participants and 2 are the confederate. 1 plays very nicely towards you, and the other 1 plays very unfairly to you. And then you give an electroshock to the 2 players. You will see that the activity in the insula and the CC is a bit reduced for the unfair player. And then when you have males instead of females playing together, you see that this reduction is much stronger in the male group compared to the female, suggesting that simply a simple game can manipulate how much we activate this region. Being a fan of the same or a different team does the same, race does the same. I don't want to go into many details, but every single difference that comes to our eyes, and then that if that is enough to create a group, that is also creating changes in these activities. The way I relate with my kin or my group is different compared to the others. And the same is true for the responsibility. So if I'm the one that is actually going and hurt you, I come and kick you, then the amount of activity that I have in the interinsula and the CC is much stronger than if I'm not the one doing that. And we tested this by having 2 participants playing a flanker task together. So basically, you need to respond as fast as possible to the middle letter, and you have 2 people playing at the same time. So by manipulating a little bit the conditions, what you can do, so you know that every time there is a mistake, she will get an electroshock. But then, because you have two participants, you have cases in which both of you are correct, so then there is no pain, everything is fine. And then a case in which the participant is incorrect and the other player is correct. So in this case, the full responsibility is of the participant, because the other is correct. So the participant is causing the pain of sweating. And then you have a case in which both of you are incorrect, and in this case you have a share of the responsibility, and then you have a case in which the participant is correct and the other is incorrect, and that means that the other has his own responsibility of receiving the pain. And when you do that and you measure the brain activity, you do see that the strongest activity is when you see the pain of others and you were the responsible for it. Sharing the responsibility already reduces the level of activity to the level of which that you have when there is no responsibility at all. And playing around again with inter-individual differences, we thought, okay, what is happening in this case in which people like psychopaths really have no sense of guilt when they commit crime? If this system is kind of in there to help you to get a perception of what goes in the other, and it somehow helps you also in your decision-making processes to take a certain decision rather than another, so you would not just go around and kill ourselves. And that is also not happening in the animal world. I mean, there is probably more violence, but it's not random. It's not like that animals and humans just start killing each other like this for the fun of killing. Not in the majority of the cases. So what is going on in the head of these people, right? Because they see you. Would they not just perceive your pain? Would they ignore it? It's just a cognitive process, or the activity in these areas is also reduced. So we had the opportunity to have them coming in the lab, and we tested them by letting them watch videos of this type. One case we created a painful condition. They're not very small. And then in the other case, we had a condition that was social kind of pain. It was exclusion, so an experimenter would come towards you and he would just push it away, and people really perceived rejected. And then we had a sort of a neutral condition where two hands just move, and final, a pleasant one. Sorry for the movies, but. And then at the same time, we made them experience the same. We hit them, we excluded them, we shake hands with them, and we caressed them. Right? And the idea was to look for, indeed, boxes that were common to both and to see whether they were reduced in them. So the first thing that we actually did was to test whether there was any difference in the experience of this stimuli to start with, because some people would argue that they do experience pain in a different way than normal controls, but that was not the case. Therefore, in our group, there were no differences in the way the brain reacted to the first-hand experience of these emotions. And when we look at the overlap, we see that in the healthy control and in the control group, we had the circuits we expected to be there. So the somatosensory, the premotor, the insula, and the anterior cingulate cortex, because they are all involved, right? In our stimulants, there's emotions, there is emotion, there is sensation, all of it. And when we look at the activity in the psychopath, right, and we compute the difference, we see that psychopaths were significantly activating these regions less. So at first we conclude, oh well, that's exactly what we thought, right? They activate this region less, and this is probably why they have an easier time to be cruel. But I was also hearing a lot of stories like, oh, there was such a nice guy, was the perfect, the dad I would imagine like my husband to be. You know? You hear all these stories. And I thought, okay, then there is something a bit weird, because if the activity in these regions really help us to get an insight in the other, then if you behave so nicely, you should be able to understand what goes on in the other, so the area should be there. So why we don't find it, and why do you have these reports? So we asked our participants this time explicitly to put themselves in the shoes of the receiving hand. So we presented the stimuli again, the painful and the caress. And when we do that, so when the psychopaths were asked to put themselves in the receiving hand, the difference that we observed before was gone. So we interpret these results as suggesting the psychopath, they probably do not lack the ability to empathize or to activate these regions. But what they do not do is to activate them spontaneously. Probably they are able to regulate them, or we don't know, actually. I skipped it. So if we now plot, I'm trying to include this bidimensional description of each of us, basically. And if we only plot here empathic ability, right, we would end up placing ourselves or empathic individual at the very far away of this line, because here we score very high. And we would end up placing the psychopath here with very low value, and then conclude that they are not empathic. But then if we now add a second dimension, which is the propensity to activate these regions, then we would place the empathic individual probably here, where they have the ability and they also have a strong propensity to do so. But then the psychopath would then be moved here, where they have the ability to do so, but they would not do it spontaneously, so they probably don't show propensity to do so. And this could inform also other conditions, but importantly, even traditional therapy, because many people now try to teach psychopaths to be empathic. They go through empathy training, which might not be necessary. It might, but it might be not. Because if they can, teaching them how to be, well, they already know how to do it. What we need to teach them probably is how to transform this into a habit, to really make it, to give them enough motivation to do it normally. And the last slide of my talk, I want to give you also the idea that this regulation of the activity of these regions is really within all of us. So what we did here, we made participants to watch movies. We were very inspired by Ure's work. So we showed them The Champ and A Perfect World, and these movies are very emotional, and they include positive moments and negative moments. They really present nice extremes. And we asked our participants to watch them in 3 conditions. And 2 are really relevant for this talk now. One is to really empathize with the kid, and the other one was to take a distance, like try to imagine to be indeed a scientist that wants to measure everything in an objective way. And when you do that, and in this case, we also run into subject correlation analysis, we see that the brain between participants becomes more synchronized in the empathic condition. So suggesting that indeed, if you upregulate these regions, you can attune better with the others. So we have many cases in many contexts in which we do need to regulate. I mean, we can't simply let this activity always be nice. So we need to have this ability to regulate. And what it might happen is that in psychopath or in more extreme cases, you have also an extreme regulation of these regions. So here, to sum up, we do have activities in the brain that indeed seem to give you help to have a perception of what goes on in the others in terms of emotions, actions, and sensations. And these perceptions really might also help, or with the decision-making, enter the decision-making machinery together with many of other information, but still might help you to select one behavior compared to the other. And this is not a black and white story. The context, inter-individual differences, everything is now playing a role and modulating this activity. And what you need to do is to find the right balance, or the balance that is culturally accepted. We can also see it that way. So we need to find a balance, and once this balance, which could also be like a statistic result, then when you find this, then you would act in the conditions you are in. And with this, I would like to end the talk and thank all the people that participated in the work. Christian, of course, with whom I enjoy science a lot, and we had this crazy idea to lead a lab together, which is great fun, by the way. All the funding agency, and then Abdal and Judith for the TMS studies on action perception, and also Sara, Alessio, and Nicola for the combination with fMRI and TMS, and Abdal also for the weight discrimination task. All the people of the lab now that are following up the prosocial behavior and its implication, and in particular Harma for the work on the psychopath, which was very long and interesting work, Sway for the responsibility, and Selene for indeed the whole PhD work on the prosocial behavior. And finally, thank you all for listening.
Thank you. Thank you very much for the question. Are there any questions?
Yeah.
Yeah, that was a wonderful, wonderful talk. Thank you so much. I don't know so much about TMS, and I just have a question that may be too specific for TMS, but you showed what happened when there was activation in S1. Is it possible to have the TMS over the hand area versus the foot area, so that when you're trying to weigh something by hand, or even in your other experiment of caressing or hitting, that it's body part specific, or is it all S1?
No, I would expect some specificity over there. We were actually more interested in checking the face area versus the hand. We didn't do that because the face areas is more ventral. So when you TMS there, you also have a lot of muscles and you twitch the muscles as well. So it's quite annoying. And then you induce a sensation. Then you don't know anymore what you're looking at. We didn't think of the foot because we didn't have the stimuli with the foot, but it could be a perfect control. And honestly, I would not expect the same results. Because when we really played around a little bit, it was kind of specific to the hand, and the fact that it only acts on the hand also suggests that there is something. But yeah, I agree. And it's, yeah.
So, thank you very much.